Okay, I think you guys should be good to go.
Yay!
Hello, welcome everyone to the Live with Nassim. This is going to be somewhat of a interesting Live with Nassim because we don't have Nassim with us, but we do have the wonderful and brilliant Dr. Inés Urdaneta. How are you, Inés?
Hi, William. Thanks. I'm fine. And you, how are you?
Oh, doing great. Yeah. No. Really looking forward to getting into some discussion here, even though, you know, Nassim was unable to join us here. He's got some very important work he's doing for, you know, the advancements of the technology here. I'm here at Taurus Tech, the main laboratory with Nassim and other research scientists. Where are you joining us from, Inés?
I'm in Mexico City.
Mexico City, excellent.
Yeah, Mexico City. Yeah. We're. I think in a couple of weeks, maybe next month, I will have another like laboratory, you know, for the Shinterji experiments. It's already set. Yeah. Well, we had a little bit of a pause because of what happened in between, but now we're starting like again the set of experiments in Shinterji. So
Oh, fantastic. Yeah, well, for anybody who doesn't know, Inés and the team of researchers she's been working with have been doing consciousness experiments. And actually, we've been doing it as a collaboration because we've done some forms of the experiment that her team is doing. We've done some of that here at Taurus Tech, and I got some really interesting results. But so last time I saw you were in the process of setting up a whole new testing methodology. Yeah. Okay.
Yeah. We were trying to create a kind of Faraday cage, a very rudimentary, because of course the advantage of doing the experiments over there in Taurus Tech is the most advanced technology you have over there, and the Faraday cage, which is, you know, this it's like a room, it's isolated from any electromagnetic radiation, it's all in copper. It's my feeling that it's all inside of copper, and then it's got some isolation from noise as well. So it's a very amazing thing. And the laser, let's say the laser beam you use in Taurus Tech is not a laser, in fact it's just a photon emitter, but it's so sensible, the photomultiplier, the detector. So it's a very different experiment from what we are doing here in Mexico. And the interesting thing is that you have, so they complement to each other because of the regime of light we use, of the frequency and all that, you have like complementary information. So for the ones that don't know about this experiment, this is a person who's trained in suction training. It's like a Buddhist, it's like in fact, it comes from the Bon tradition, which is prior to the Buddhism we know now. So the Bon tradition is the one that's been followed, and the suction in particular. And then this person who is like our subject of experimentation, he enters into these states, which we call Shinterji states, because it's following a certain methodology, which is a Shinterji model. And then we detect if there's any change in the light emitted from a source when you detect it at the other side. So you detect the photons at the other side, at the detector, and we try to see if there's any influence of this consciousness state over the detection of the photons. So that's basically the experiment we have been running over there at Taurus Tech and here in Mexico. And so we have been having very interesting preliminary results, but of course, this is just in a very preliminary stage. So we have to keep, in order to present this like publicly and like say, okay, this is what's happening. We really have to keep doing more experiments. So yeah, but basically, so we try to create a kind of a similar Faraday cage, which is basically putting all the setup inside pipes. Isolating inside copper pipes. So yeah, it's looking nice, but we still have to see how much of an isolation that is. But the good thing with the laser field, because you know isolation was very important when we were using the light source over there at our stack, because it's extremely sensible. You know, we could see the sensibility of this photomultiplier. It detected the order of photons. Meanwhile, here we're using a photoresistant. It's very insensible. It's very, so if a laser is a very strong light source, it will be very difficult to have contamination like from other light sources because of the kind of detector that we have that is very insensible. It's a photoresistant. So if everything is really dark, you can see that there's basically very small contamination. Meanwhile, in the Faraday cage we were having at your experiment at your place, it was really, the contamination was so easy. Do you remember that?
Oh yeah, yeah.
Yeah, even photons from your body could be accounted for.
Yeah. The smallest little photon source, and it's registering big time on the photomultiplier tube here. Yeah.
So that's different things of setups. But yeah, I think like for next month, maybe we'll retake this part. Yeah.
Oh, fantastic. I'm so excited to see more data on that, because it's just so interesting. I mean, you know, clearly showing an effect on the photon flux with the meditator there next to the apparatus. But even what I find extremely interesting, when the effect is projected into the future or the past, you see an effect still. And that's one of the most exciting aspects to me, because it kind of shows this transtemporal influence across time of the consciousness.
This non-locality,
Non-locality.
Well, we have to be very, very careful with that, right? So this is why we have to reproduce experiments as much as possible and be really sure that that's what's happening. Because of course, the extraordinary, just as Carl Sagan said, that extraordinary claims require extraordinary evidence. The other way also holds true. Extraordinary evidence requires extraordinary claims. So we have the other way around, and we have to be really careful. So but that's true. We even saw some kind of effects when we saw the people from Viva Vision. You remember?
Oh yeah.
So, but that still has to be analyzed.
Oh, very exciting. And we've got some experiments underway here. I had mentioned on the faculty Q&A that we're starting experiments with the arc resonant kinetics crystals, the arc crystals. We're starting experiments, third-party independent testing at the university with Dr. Lydia. And she's going to be doing testing with the arc crystals on cells, human cells, in vitro. So this should be very interesting, because this is an experiment that I've been wanting to do for many years, because you know, we've used plants as our model organism, where you know, we can give them water that has been treated with the resonance technology, the arc crystal, or you know, seeds that have been grown in proximity to the crystal. And, you know, we're able to see responses in the plants as indicators of, you know, an effect on the biology of the technology. But to be able to do it with isolated cells, where we can go in and look at things like the gene expression, metabolic markers at the molecular level, this should just be a wealth of data for us to be able to understand exactly what effects are occurring in the biological system, down to the DNA and mitochondrial level of the cell. And that's being spearheaded and undertaken by our colleague here at the Residence Academy, Dr. Lydia. But that should be very interesting, and those experiments are starting in the next few months here. So very much looking forward to that as well. We have a conference coming up. There's the 2021 biohacking conference. I think it's the 7th annual biohacking conference. And, biohacking is this term that has popped up, that just kind of is this view of how to optimize your body, how to optimize the biological system, particularly using the science that has come to the fore. And this includes things like mindfulness practices, exercise, health and nutrition, eating healthy foods, this kind of stuff, but also technologies that can assist and augment those practices for bio-optimization. And so we're going to be there showcasing the resonance technology, specifically the advanced resonance kinetics, arc crystal technology. So if anybody's in the area, it's in Orlando, Florida, you can try to maybe swing by. We'll be there, and you can see some of the latest products and science and innovation that Arc Crystal is coming out with some really great stuff. I think that the water bottles are available now, and this is a really nifty way to get your Arc Crystal, and there's a indentation in the bottle so that you can get the Arc Crystal positioned perfectly to potentiate the water. Yeah. So a lot of cool products and innovations and results from experiments that we've been doing that are going to be available.
Yeah. I was seeing now there's like a kind of, now that you were talking about biology, there's this question from Oriana, which I think would be very interesting to address. So maybe, I don't know, we should do it like we usually do, asking the people to come into the call and ask the question directly. I don't know if Jamie or you're hosting but you're the host now, right?
I think I'm a co-host, but we'll just tell them the name, and if they can, I'm sure they'll bring them on. Okay. And so this is Oriana. Okay.
Oriana, yeah.
Okay, one sec. You can go ahead and start answering it. I'll bring her on.
Okay, so the question was, cells within the Sinoatrial Node are the primary pacemaker site within the heart. These cells are characterized as having no true resting potential, but instead generate regular spontaneous action potentials. Can you explain this in biophysics, and how does this relate to the first heartbeat of the embryo 1 month after conception? Wow, that's a
Thank you for the question, Oriana. Very, very interesting. This is a particular area of study for myself, because the sinoatrial node, I believe, is very central to the biophysics of the body. Obviously, because it is the pacemaker of the heart. But also for some not so obvious reasons that I've been investigating for several years now. So the sinoatrial node is a very interesting organ of the heart, structure of the heart. It is almost like a little gland, about the size, a little bit smaller than the pinky. And it sits above the right atrium. And what's interesting about it is that it generates a electrochemical action potential that is the release of an electrical signal across the right atrium into another node that signals the contraction of the ventricles of the heart to produce the pumping action. So this is the heartbeat, the source of the heartbeat. And it starts with this node of specialized cells. And so some of the interesting things about it is that it is initiating and firing this electrochemical action potential independent of the electrical activity of the central nervous system. So, we generally think of all the, electrical signaling activity as being localized in the central nervous system and the skeletal muscular system. But this electrical signaling is totally independent of the brain and the central nervous system. And, this is part of what has even been likened to the brain of the heart. The heart's own neuronal-like signaling system and electrochemical action potentials. A very interesting thing about the sinoatrial node is that I find some parallels in structure to like the pineal gland, in that that little node kind of forms a shell that's hollow in the center. This is much like the pineal gland. What's interesting is these are both at energetic centers that have been likened to, in terms of the philosophical, energetic description from Eastern philosophies is chakras, chakra points. And so these, for me, are kind of like the physiological correlates of where these energetic centers have a real physical structure and organ associated with the energetic dynamics of things like chakras and meridians. And one of the thoughts that we have entertained and explored, where, in these centers, you have a very high amount of energy being produced and exchanged in these areas. This is generating the electrochemical action potential that is driving the contraction, the beating of the heart, and that hollow spherical shell of cells. It's very interesting to entertain the idea if perhaps there is like a singularity in the center. So that's where like the unified physics starts to come into. So this might be like the singularity point of the heart. And, it's where the orchestration for that beating, that contraction is occurring. And, we can see parallels in structure in other places of the body that happen to correlate with energetic centers and important physiological locations, like the pineal gland as well, which might be the singularity point of the brain, where you have at a quantum level, at the microscopic scale, a literal singularity that is involved in the energy and information exchange occurring. You're muted, Oriana. I don't know if we can hear you.
Oh, this is probably a good thing. I'm getting really excited. But part of it in the second part of my question had to do with the heart rate variability, but also this of when that first heartbeat, that first contraction, and what the most recent that I've seen is that I was asking about the mitochondria from the father, because of course it's only inherited through the mother, which means it's in the cell egg prior to conception. but what happens to the mitochondria in the flagella of the sperm. And it turns out that the head of the sperm pops open, out comes the genome, the 23 chromosomes from dad, but also a centriole. Now, this centriole became really interesting to me because that's the command center for the mitochondria. And what happens then for this orchestrating the cell division appears to orchestrate prior to the genome then recombining, i.e. conception. So this was like, to me, I just heard the words immaculate conception, like, whoa, what's orchestrating this command center for the mitochondria, which is the communication in the primordial water. And the centriole is a series of 27 tubules, microtubules, that contain atomic water. Now that water then is being pulsed in this encoding. And what I'm wondering, and I haven't seen anything further about it, is if that then becomes that SA point, that cyanoidal point, as the embryo unfurls and unfolds, that it's pulsing until those mitochondria contract. And there's only 5 or 6 pacer cells that begin the contraction of the heart muscle at 1 month after conception, or 29 days, I believe it is.
Yeah.
I also believe it's 1 full cycle of the moon, that there's a celestial rhythm that is that pulsing of that first centriole that pulses the mitochondrial, that then pulses the heartbeat.
Well, the centriole is the nucleation point for the microtubules. And then, of course, the mitochondria, they form helical filaments along the microtubules. And this is called the mitochondrial reticular matrix. But then, the centriole is absolutely vital for organizing the chromosomes for cellular division. So, that first centriole in the embryo, the fertilized egg, is going to be responsible for orchestrating and ordering the first round of cellular division, and then, getting to the multicellular stage of development.
And the centriole is outside the cell and inside the cell. Is that correct?
No, the centriole, it locates right near the nucleus of the inside of the cell. It isn't. It doesn't span the cellular membrane.
Oh, okay.
Yeah. So it forms.
Does it have a connection with the biophoton field?
It does. So it forms the nucleation center. So it's kind of like the central organizing location for the cytoskeleton, for all the microtubules. They kind of radiate out from the centriole. And then, of course, the mitochondria, they're always, nearly always very integrally connected with the microtubule network of the cell. And there you have the majority of biophoton production and channeling. And actually, that biophoton production kind of relates back to the idea of that first heartbeat, because one of the reasons why that's kind of such a pivotal moment in the development of the fetus is that once you have the sinoatrial node firing electrical action potentials, the heart is then sending out electrical currents through the body. Even in some of the earliest stages of embryo development, you have electrical gradients that are being formed. And one of the reasons this is so important is that these electrical gradients are the veritable morphogenic field of the organism. So the morphogenesis, the development of body patterning and structure is templated on electrical currents that are running through the organism. Now, once you have the electrical currents coming from the heart, those electrical currents are strong enough that they're going to the membranes, the plasma membrane of every cell in the organism. They're traveling along the microtubules, exciting them for things like biophoton emission, and they're being channeled into even the DNA, which can conduct electrons as well. So this kind of becomes like an electrical animator. It's an animation center for the organism, where the electrical currents being generated by the heart are passing in coherent waves, coherent pulses, electrical currents through not only the tissues of the body, but also into the cells, into the nucleus. And those electrical excitations can facilitate the coherent dipole interaction of nucleotides in the DNA, tubulin in the microtubules. And so this becomes very important in the information processing and exchange via electrons and biophotons and phonons in the body.
Right, and that pulsing it through the water, would the water then be electrically charged and also capable of carrying the geometric pattern, much like a sonar wave with the dolphin has an interference pattern that is reading the shape, size, sight, but also the textures of what it's encountering. And could the water of the cell actually be communicating through this interference patterns carried in the electrical current?
Well, so in particular, the polymer molecules of the cell, like DNA, like tubulin, these all have electrical dipoles, which are basically, you've got ring structures of the molecules that have a cloud of free electrons. These are called pi delocalized electrons. And but there are electrons that are similar to the electrons in a metal, which are conductive, because they're delocalized. So they're able to move around a little bit more freely than the electrons that are bound in like covalent and ionic bonds. And so the DNA is a polymer molecule. There's just a stack of these benzene rings with these delocalized electrons. So it's a stack of all these dipoles, because those free electrons can oscillate back and forth. And what that allows it to do is for the water, the interfacial water, to be coupled to those dipole oscillations. So that's where you're having the coupling occurring, because water has a partial dipole moment. So when you have those electrical currents passing through DNA, microtubules, mitochondria, it's interacting with those dipole moments of the macromolecular structures, which is coupled with the partial dipole moment of the interfacial water. So that's where you're getting this strong coupling. And the water is able to act as the intermediary between different molecules to exchange and coordinate this information. So for instance, if you have a gene in the DNA and you have a, say, transcription factor that's going to initiate transcription of that gene, it's going to bind to the DNA. The way that it reads the DNA inside of this helical phosphorus backbone, the way that it reads those inside dipole moments of the nucleotides is via the structured water intermediary.
Right.
Yeah.
So that it's following the blueprint, but what's the intelligence that's actually creating this coordination, communication? And one thing I want to come to, because Dr. Inés got me on to my whole question about the vortex of energy and knowing the heart creates a vortex of energy. But what I was feeling is I'm exploring this internally. I'm trained in remote viewing biochemistry, and I've trained myself to go internally to feel the sensation. So inside this synodal, the SA point is where I'm beginning to track this vortex, but the vortex kept wobbling, and then I realized, oh, what if it's a double vortex, that it's spinning at a moment that it spins in spirals, and it's a delicate dynamic balance of a still point and all movement simultaneously. And what of that movement then creates the double torus that creates the counterspin. Now, counterspin comes back to what you were just saying, William, about the electrical current being read in the water, but what of the magnetic field? For if there's an electrical flow, there's a magnetic flow or a field, right? That it's, in other words, is that the counterspin that's creating the magnetic field that's essentially holding the electrical field in a pattern? And is this pattern then more essential, meaning prior to any biochemical configuration? The molecules form from this template, and then this template, the other thing I've been tracking is time crystals. So time crystals have as all crystals do, an ordered repeating pattern, except time crystals have an ordered repeating pattern in time. Whoa, what a rhythm is an ordered repeating pattern in time. And what if this time crystal is actually a fractal pattern, ordered repeating pattern that is pulsing, and that pulsing then creates the first heartbeat and every heartbeat. Yeah, and if that time crystal, what a great way for evolution to encode all of its learning through billions of years, 3.8 billions of years from a one-celled organism to be a multi-celled being, and we do it in 9 months. Whoa, what's that rhythm, pulsing pattern, unfurling, unfolding, that is still pulsing pattern within each of us? And can we come back into that higher coherence, his heart math? I just was studying with Roland McCready in heart math, and they really have found that coherent rhythmic pattern, but also that they can predict someone's age within 2 years because their variability lessens as they age. So I wondered what lessens, what diminishes as we age, and what do children have this delightful wonder of the world that is heartfelt. Whoa, what if the heart can feel wonder? Oh, I wonder what that would feel like. Wow. Wait. Those are all my explorations of, but I'm quite sure that double helix is within the heart, and that's part of our evolution, is actually to enter and activate it, and consciously, that we have to consciously come into.
Can I ask a question? Because now that, wow, this was overwhelming. Okay. From what I have, okay, so there's some studies from Maria Estel del Rio. She's a Mexican, she was active researcher some years ago. Now I think she's retired. But she was one of the first ones to find that in cells you would have crystal water. It's like, how do you call that?
Liquid crystalline?
Liquid crystals, yeah. She was the ones to find the first to find these liquid crystals in the cells, which is first very important to see, because that's the center of memory. That's the part of the cell that is also encoding information and registering the information as well. But she also found that in what we call the chakras, there's a very interesting amount of magnetite in these places. So I was wondering, because that's the question I wanted to ask you, William, if we know there's a center of magnetite inside the heart, for instance. No, I don't know. I didn't, because her study clearly shows, and maybe I can pull out the link afterwards. It clearly shows how these magnetite concentrations are larger in the chakra points, let's say. And this would be the glands, basically. So basically in the glands. But I was wondering, I guess it would be reasonable to think that in the heart, and because of this magnetism that you were talking about, Oriana, there would have to be some kind of concentration of magnetite, which is one of the questions I was having. It also made me think that when you have a singularity, so if I understand well, William, the singularity would be placed in this particular place that you mentioned, which is that makes perfect sense. But as I see the singularity, it's always a double torus dynamics, because if you don't have double torus, you don't have singularity. It's exactly because there's this double, yeah. So they're synonyms. You have singularity, you must have always a double torus dynamics. If you have a single torus, there's no singularity. There cannot be. It's a problem of the topology of the problem. So then, if you have double torus dynamics, another thing that that's part of my exploration in because I'm seeing the PSU in my research as a quantum of consciousness. So in the things I'm developing, and that's why I've been doing this collaboration with a team here in Mexico, and we're working on that concept, is the fact that is this double torus dynamics is this, which is a feedback feed-forward mechanism as well. It's present from the Planck and the sub-Planck up to the cosmological scale. It's always there. So and it is a nesting of this double torus dynamics. This is a nesting of this dynamics from the sub-Planck to the Planck to the universe. So my first thought about it is that in this pulse we have in this of the heart, the beat of the heart, all scales are implied, because so then in this heartbeat, every scale is participating, from the very small to the very big. They're all being part of this heartbeat. So on one side, and then and the other aspect of this oscillation of the double torus dynamics, which is, in my view, a quantum of consciousness itself, because it's a feed-forward unit, let's say. So it's a feedback feed-forward unit. So that's why I call it a quantum of consciousness. So that's part of an elective I will probably do at some point after Model 8, I hope. So so then the thing is that you would have to think about it like in this dynamics of this PSU, this double torus dynamics, electricity, magnetism, they're all parts of it. It's not that one created the other, they're simultaneously happening. It's like they're different aspects of the same of the feedback feed-forward mechanism. So if you're talking about magnetism, you're referring to one of the aspects of this feedback feed-forward mechanism. If you say matter, then that's another aspect of the feedback feed-forward mechanism. So they're all emerging from the feedback feed-forward mechanism, which is at the root, at the base of this of all PSU and of everything of life. So yeah, exactly.
That makes so much sense. And I had been actually calling it the feedback feed-forward mechanism, as well, as I came upon this time crystal that was, what creates that heartbeat? Where does evolution keep that rhythmic pattern? And that singularity then became, the singularity, but then it was the feedback feed-forward was all the movement, was all one motion going through a still point, right? So that still became the dynamic balance point, but how would still feel movement? In a dynamic like a tumbler jumping up around, they're always moving around a still point. right? So then I looked at all the music that ever was written out for all the instruments, for all the eons, and they all have one thing in common: the rest in between the notes. It's all the same rest, right? So that still point became all the same still point. There's only one. Whoa, well then what allows it to flow through? And that feedback feed forward became spin, counter spin.
Yeah,
And that spin, counter spin then from a sub-Planckian unobservable universe then became observable, but it's all the same spin, counter spin.
Yeah, it's always the same.
And somehow that's what I was getting to with the magnetic field. There's always a magnetic flow with the electrical current. So what of the magnetic is actually just being created from the counter spin that is unobserved, and yet creates the inflowing of the, and then it became a masculine, feminine, the yin yang of the yin yang symbol that spins into a double torus, right? Where the masculine or the male polarity is then the outflow, where the female polarity is the receiver, and that it's receiving in the counterspin, where rather than it being antimatter and exploding with each other, they're actually merging and balancing, but they're merging and balancing at all levels.
Yeah, yeah, that's lovely. Yeah, and yeah, because when you see the heartbeat, it could be seen like breathing, like a contraction and an expansion, contraction and expansion, just as the cube octahedron, when you fold it and unfold it, it's basically the same dynamics, and it's always this counter spin, counter spin, spin, counter spin, which is yeah, at the basis.
Right. Okay, so real quickly, I'll let you get on, because I'm just starting to work now with a couple of MDs and a psychiatrist and a psychotherapist, and we've got a group forming to just feel into this heartbeat, the variability itself, which is where I'm feeling this, that original beat of the heart that we feel as an embryo is still within us, and it's inherently knowable to come back in coherence with what's that unfurling, unfolding pattern of evolution, and what's the entelechy of evolution. As an acorn has the entelechy to be an oak tree, what is that undulating, underlying, unfurling pattern that we can still feel and come into coherence with? Okay. Thank you all.
Thank you. Thanks, Soriana. It's beautiful. But maybe we could link that question, because you had a second one, which is related to the Heart Math Institute.
Yeah, that's just what I was bringing up, that idea of that variability that they're measuring. I'm convinced it's a language. It's a universal language that is pulsed in a rhythmic, unfurling, unfolding pattern, and that we can feel it and sense it and actually become sensate as the language of sensation. Okay.
Yeah. Okay. Thanks.
Okay. Thanks.
I think that this question connects really nicely with Evelyn's Moyer's question, which is, I get that consciousness is a feedback mechanism between the external and the internal worlds, but what about the subconscious or the unconscious? Yeah. Yeah, maybe we can address that, because it makes part of the, yeah. So
Who's that from, Inez?
that was from wait, that was from Evelyn Moyer.
Okay, go ahead.
Yeah. So because I like this question, because I always have it. Also, I do it myself, because when we talk about consciousness, and this is a discussion we have always had also with William and Nassim, and this because for me, when we talk about consciousness, like the image I have is the one, like the one we have. Hi, Evelyn. You're muted. Yeah.
Hello.
Hi, hello. Hello. Yeah. Okay, so I'll start and maybe then I don't know. So the thing is, when I think about consciousness, I have this analogy with baryonic matter. The things that we can see is what we call the mass, the baryonic mass. But then physics realized that there was a lot of information energy that was missing, and we call it dark energy and dark mass. And that's something that's been worked in detail in Module 7 in order to understand where is it really this dark mass and dark energy. So I like this analogy in which this dark mass and dark energy, which is really energy and of the vacuum properly accounted for, its spin and its dynamics, that's what accounts for that part, is what I would call the unconscious as well. Because from the reference frame of our perspective, which is not able to perceive everything that's happening in all these dynamics, no, we only see the tip of the iceberg, which is this baryonic matter, which is what we perceive as an object, and that's what we call, like, in my in my perspective, that would be like the conscious self, the one that can see what is happening, but that's only the tip of the iceberg to what all that's going on below, which is all the information exchange dynamics among all the scales and all this feed-forward mechanism, which is, at this moment, I'm aware of partially who I am, but I have no idea of what's happening inside my body, all these internal processes going on, the amount of energy that's being expended and exchanged, and the information coming in and coming out. And that all, for me, is part of what my unconscious self is, which is based in, and that's why I like so much this analogy, because in physics, we have the same analogy. There's we thought everything that existed was that we saw, the matter that we saw and all that, and suddenly we realized there's a lot more of mass and energy that we don't account for. And where is it? Well, that would be like my analogy with the unconscious self. So for me, the consciousness, that's probably the limitation we have because of our senses and because we're limited in space and time in a certain frame. So because we're in these conditions, then we are limited to this amount to this knowledge of the information. Maybe for the universe itself, it's not unconscious at all, because it's got knowledge of all the processes embedded within. But then we have this philosophical question: is the universe? How does the universe know that's all that there is, because it's not a closed system. So yeah, so the question is open, and that's a question that many, not only philosophers, but the Vedas, and they all try to answer the same question, and, because of course, how come, and it's a beautiful inquiry. Yeah, so that would be like, so I always have that in mind. For me, unconscious is where the real issue is happening. It's the 95% missing dark mass, dark energy. That's the unconscious in my perspective. But of course, from my perspective, that's not novel for me, and maybe for anyone. But who knows for the universe itself. So, but I do believe that we share that in common with the universe. I do think that the universe, if it knew everything, it would be a closed system, and then it would not be possible. That's my and since the universe is growing and learning about itself and experiencing everything there is through all the creations and manifestations within, then it is something that it's unlimited. So, well, that would be my my experience about it.
That makes sense.
Yeah.
And you also have to consider it in the terms that the way that terminology is used, consciousness, subconscious, unconscious, that comes from the field of psychology. And so what it's really talking about is that the recognition that most of the intelligent processes, what I would even call sentient processes, are not in our waking awareness. The vast intelligence and really sentience of the body is actually much deeper and below our waking awareness. And that's what Inés was saying, that, what we call consciousness is the tip of the iceberg. And that really is that this is why consciousness science has a lot of confusion or obfuscation to get through, is because the terminology isn't even worked out yet. It's not even delineated yet. Because when we say consciousness, it seems like everybody has a little bit of a different idea of what we're talking about. And I think one of the most common thoughts in terms of when somebody refers to consciousness is that people think about their narrated experience, their cognition, the story that is told about experience. But that's actually the smallest fraction at the tip of the iceberg of what consciousness is. I have introduced in works like Unified Physics and the Entanglement Nexus of Awareness, one of my publications. And I believe we discussed it a little bit as well in the Unified Space Memory Network. It is a much general or broader definition of consciousness, and that when we refer to consciousness, mostly what we're talking about is a basal level consciousness, which is not self-awareness, which is not cognitive consciousness, which is associated with story, a narrative, and ego. Basal level consciousness is kind of like a pure awareness. And you can see this in things like the Bon tradition. We mentioned that a little bit in your Shintoji experiments, where the goal of mindfulness in the Bon tradition of Buddhism is to get rid of that narrative, that narration, that ego, the storytelling and the talking inside the head. Get rid of that and get to the basal awareness, which is called like pure awareness, clear light. It's when that kind of higher cognitive level functioning quiets down enough that you enter into the more expansive, more general, pure awareness, basal awareness, which isn't categorizing things and dividing things up and making distinctions and comparisons and judgments. It's just experiencing. And that experience, if gone into at a deep enough level, is without distinction, without division. And eventually you get to a point where there is no I in the experience. If you're in a forest and you hear the wind go through the trees, that is you. You experience that as you. You are that sound. You are that bird chirping in the distance. That's basal awareness. But to bring it back to kind of more of a scientific kind of delineation of the question, is that so that basal level consciousness, which for us mostly resides as unconscious and subconscious, because we're focused totally at the tip of the iceberg on distinctions, judgments, stories, comparisons, all of that cognitive
Identities. Yeah.
Identities, all of the cognitive activity that characterizes humans. But that is very much a result of the information feedback feed forward dynamic of the Unified Space Memory Network. Going back to, okay, so psychology realized the vast majority of intelligence and sentience of us is occurring below our conceptual awareness. It's like the body's intelligence, the cell's intelligence. If you get down into like the subcellular regime with what the mitochondria is doing and the DNA is doing, it is like unbelievably brilliant engineering nanomolecular engineering and subatomic rearrangements. And I'm definitely not cognitive of the nanomolecular engineering and subatomic rearrangements that are occurring at that level, but it's doing it with precision that is beyond belief. So I mean, the intelligence at that level is astounding. And you know, so it's just that what we call unconscious, you know, that is very much a part of the information feedback feed forward dynamic that is occurring constitutively all the time with the morphogenic field, giving the drive, the impetus for order, increasing coherence and organizational synergy. And so you know, that morphogenic dynamic, that evolution of matter and energy in the universe, it has a sentient component to it. It has an intelligence to it. And the importance where we can avoid a hang up is that we're not necessarily equating consciousness, awareness, or sentience with the higher level cognitive awareness that is normally attributed to consciousness. You know, it's not like that atoms are talking to themselves and telling themselves a story of how they're going to come together to form a macromolecule. Not that kind of intelligence, but there is an intelligence and a sentience and awareness there that's part and parcel of the dynamic. And that goes across scale from the ordering of the Planck plasma to atoms, molecules, macromolecules, cells, tissues, planets, solar systems, on and on. That you have that same sentient, intelligent dynamic at work in the organizational synergy.
Yeah. And I think with meditation, we can begin to quiet some of that story and yeah, get down into some of that basal. But I think also gratitude. If we just express our gratitude.
Very much so, yeah. Yeah. I guess, I guess even in so much as gratitude is a feeling of acceptance as well as gratefulness, but and you know, that feeling of acceptance definitely, I believe it helps you to kind of get out of that more judgmental, divisive cognition, which takes you kind of back into story mode and narration and ego. So I could very much see that, you know, feeling gratitude and thinking about, you know, appreciation kind of gets you into like a flow of kind of like acceptance. And I could very much see how that gets you into an energy space that's very conducive to get into a more expansive basal level consciousness.
Yeah, I agree. Thank you. Thank you
Thank
so
you.
much.
Thank you, Evelyn.
Thank you.
Let's see here. There's a question regarding, okay, so if we follow here, we have Jeff Colebrook question, which is related to the one about heart connection and mind, which is like following, or maybe, there's a question about the art crystal. Wait here, Patricia Sobogar. Yeah. So she's asking regarding the charging. Well, first Colebrook, yeah. Jeff Colebrook. So he's asking, how interesting the question. I was going to both. Yeah, hi.
Hi.
Hi, Jeff.
I was just commenting that I had that question in my mind, and then you did that amazing answer about the heart. There wasn't really much left for my question. I mean,
Oh, okay.
it was the answer. But it was sort of the spiritual side of you did more some of the technical, but if you want to focus a little more on the inspiration from the science for the spiritual, that would be fun too. So if you want to tie that technical, beautiful, I was just like blown away. And Oriana is amazing anyhow. But yeah, if you want to tie some of that into, I mean, you hit the singularity part of it, which was like, because so much is going on, like the heart math, everything around the awakening and the spirituality. Everyone in all the spiritual circles is talking about heart coherence and meditating in groups. That might be an interesting question, is expand, is how can we use this information to connect non-locally in heart coherence, or wherever you want to take it, if you want to add anything to it. But what you already shared was pretty powerful.
Okay. Okay. So when I was at some of these hard math, let's say collective experiences, so I noticed that. Well, I met. I don't remember his name. It's one of the first persons in hard math. He was giving a conference here in Mexico, and so we had a panel together. And it was funny because there was a third researcher. His name was Carlos Orozco, and so we were us three at the panel, and we could feel. So this other person whose name I can't remember, it was on the other side. There was then Carlos, and there was me. We were sitting before that we were called to go to the podium, and so I could feel that this person to my right, he was really like completely like out of himself. I would say that, like, he was not he was in anguish. He was really excited. It was like, so I could see that this other person, I cannot believe I can't recall his name. He was, so I didn't do hard math before. I was just learning at that moment, and so we were together beside this person, beside Carlos, and like we tried with our hearts, like entering in our hearts, in our rest, in our breathing and all that, to calm him down, because it was completely, uncoherent. It was completely incoherent. So this other person, he told Carlos, okay, let's breathe, let's start breathing. And we're talking about a person who is really used to give conferences and all. It's just all the time like that. That's his state of being, like completely overwhelmed. So and at that point, I really felt how strong that is, that if you are in your meditation state, or it doesn't even have to be a meditation, it's got to be a practice that you do all day, so that you always have this breathing, this conscious breathing in and out, and you focus on your breathing. And only because of that, you get this centering of mind and heart. That was my experience at that moment, and that's what I've been doing after. And because of this, then afterwards, I got the opportunity to do heart math some couple of times after that, and that's always what I have experienced. And so at that moment, we did this experiment with the whole audience, and we measured. So we did that, and that's when I really saw that it was an effective mechanism to peace people and to center them and to to heal in some point. So I think that's synchronization. So that's happening because of our synchronization. It's something's getting synchronized, but this synchronization is happening through space. So because I am a center of coherence, and there was on the other side this person who was coherently centering, then you can encompass everything. Like the experiment you have you seen this experiment where you have clocks which are ticking in very metrometers? That's for the music, I don't know if you have seen them, but And then suddenly they're all in decoherence. They're all ticking differently. But then at some point they all start synchronizing, and at some point they all are going at the same pace. This is happening. This could not happen if there was not a medium connecting them all, which is the table. The table is feeling this micro, so the table is acting as a synchronizer. In this case, the space in between is doing this thing. The space in between is like this silent notes in between the notes, the silence in between the notes. The space in between is creating this synchronization. So it's happening through space, and it is a space which is synchronizing everything. So it is a collective effect. If the more people go into that state, the more people practice it internally, it will happen without consciousness. And you don't have to do it like, okay, I want to do it. And if everyone just forget about everything else and you just do it for yourself, and you spend in that state the most amount of time, if everyone did that followed that just simple instruction, with not knowing anything about the other. I don't know what my neighbors are doing. I don't know with whatever. They're all doing their life. It's normal. But if everyone did that same exercise, because the synchronization will happen because the space in between connects all scales. We know that's the theory. That's the holographic theory. So everything is going to be connected through the space in between and inside, because we're all made of space as well. So for the space, it's just a continuum. There's just a little change, phase of transition phase, but that's it. It will happen. So it's just, that's my experience with that. It's really important to be centered. That's the best way you can help.
Thank you for sharing your experience. Yeah.
Yeah.
I had an art crystal question, a real quick one, since I'm here. When of course, my art crystal levitates when I hold it up in front of me. It spins and it goes counterclockwise when I go on the right. It's in my left hand. And basically it'll go really fast and vertical, like just levitates. But when I do it with my right hand, it goes clockwise. And and I just noticed that phenomena recently. And of course, it levitates both ways when I use it as a pendulum. And when I meditate, it goes vertical. I mean, I can't hold my arms straight. So, and that's obviously interacting with my field. But I was just wondering why the clockwise, counterclockwise for left hand, right hand. I don't know. It's kind of, if anybody else experienced that or uses it as a pendulum.
It's a good idea to use it as a pendulum. I know people that do that.
I mean, it goes vertical. I mean, I can't hold it. It's powerful. It's actually tapping into gravity control. Interacting with my own field at a level. So, but thank you. And yeah, very inspirational story. And module 7 is such an incredible piece of work.
Oh, you like it?
Oh, even the art. It's fantastic. It just broke so much thinking. And I bless you all for that work. Marvelous job. Just incredible job on module 7. Thank you.
Thank you.
I'm getting near the end.
Oh, thank you.
Thank you.
I'm glad you like it. Thank you. Okay. So there's a question from Patricia Beaugerard. She's asking regarding charging liquids with arc crystals. My understanding is that the charged water can charge other liquids that it's introduced. I had charged water to my hot tea or veggie juice. What's the approximate time to let the communication structuring take place? I charge all my liquids, oils, liquid supplements. Okay.
Hi, Patricia.
You're muted.
So
There I am.
the initial modulation can happen extremely rapidly. So let's see, you're taking potentiated water, charged water, and using it with other liquids. So almost as soon as the non-structured liquids come into contact with the structured water, nearly immediately it will begin the reorientation of that non-structured phase of the liquid that you've introduced it into. So it can happen extremely extremely quickly. And we have seen measurements that show how quickly the onset of this change can occur. But, we are talking about rearrangements at the molecular level. So even though it is happening extremely quickly, because there's so much material at the molecular level, it can take what seems to us like a long time for a large volume to become fully charged. So what I would say is that you can start to see benefits from introducing the structured water to another liquid nearly immediately. But for that phase propagation, that structuring to propagate through the other liquid, I think that, depending on the volume, as much as like an overnight incubation period, that's kind of that's a little bit more on the longer side of things. But like I would anticipate that after a good 12-hour cycle, it'd be nearly fully saturated with the phase transition to a more ordered, coherent structure. But so, very rapidly it begins. So, I mean, almost immediately you can get benefits from it. 30 minutes, an hour, you're getting benefits from it. And then, saturation of the benefit, depending on the volume, at about 12 hours.
Cool. So one thing I wanted everybody to know, they could be charging all their water with our crystal, of course. But so what I've been doing, William, is say I come in with a 16-ounce veggie juice. If I add our crystal water to it and give it 10 minutes, does it have any value, or is that not worth doing?
Yeah. That's worth doing. Yeah. I guess it kind of goes without saying that the longer that you can give it, the better. But I mean, I consider that the beneficial effects actually have a very rapid onset. And part of that is just that I'm viewing it as intermolecular interactions. And at that scale, things happen very quickly. Water to water interactions are in like the femtosecond time scale. So there's a trillion of those in a second. So I mean, a trillion interactions at a slow pace in 1 second. So to us, that's pretty immediate. Yeah, but it's just that, again, so that effect is being translated very rapidly, but trillions of interactions a second. But with a large volume of water, we're talking about trillions and trillions and trillions and trillions of molecules. So that takes some time to fully propagate out and fully saturate that phase transition to a more structured ordering phase.
Cool. So I'm basically looking at pass fail, like, is it worth taking the step to do that? And what I hear you say is yes. Yeah. I know on a previous talk, somebody had asked about boiling water. So if you boil structured water, it loses its structure. Because we were talking about running it through a coffee filter or whatever.
Mhm.
So my thought is every time I make tea, I just take some of my charged arc water and add it to that because I figure I'm activating, even though it was boiled, at least that communication goes through. Same
Mm.
with veggie juice. Obviously, I'm not going to let it sit 12 hours, but I figure maybe if the time I'm talking to you, it's got arc water activating it, it's restructuring it at some level. And I hear you say, yes, it's worth doing.
Yeah, yeah, and you know the boiling though, or passing the water, through the coffee maker, that's going to obliterate any structure.
Right.
You
That's why
have
I'm
to
adding it
deliver.
after
Yeah.
the fact.
Yeah. So that's the
Like
way to
the
do it.
extract.
Yeah,
Cool.
yeah.
Yeah. And just something to pass on to everybody. Like, I mean, I charge everything. If I've got trace minerals, I charge that. If I've got like a face serum, vitamin C, I charge that. I basically charge everything. So I love my art crystal. Take care, William. Good to see you. I loved
Good.
your answer today.
Oh, thank you. Good to see you.
Yeah. Take care.
Take care.
Bye.
Bye, thanks.
Hi, Inez.
Bye. Okay, so next question would be David Knox. So David Knox is asking, evolution according to scientific materialism is due exclusively to random mutations of DNA. I love this question. Yet the form and function can only be correlated, usually loosely, to gene sequences, and instincts have not been found there at all. The morphic field is the obvious next place to look for the source of information and orchestration. So one question is, how do we disprove the idea that random mutations in an unconscious universe are responsible for the advent and evolution of life on Earth? And two, are there ways we can isolate and study aspects of the morphic field? Awesome question. Hi, David.
Hi guys, how you doing?
Hi.
Good.
Great to see
Great.
you. So,
Great insight on this question.
you know, when I was thinking about, I have a whole podcast about this stuff. When I was thinking about random mutations in the DNA and how you would prove or disprove that, well, let's just say not even random. Let's take the word random out and just say DNA causes changes. Well, you'd have to be able to take a species in one condition and then a slightly evolved species and compare their DNA and be able to find out which ones were changed, right? In order to isolate that particular thing. Let's say a caterpillar is crawling around and then a couple million years later, a caterpillar somehow evolves the ability to encase itself in a chrysalis, break its body down to goo, bring it back up into this beautiful butterfly, right? That's probably a fairly complex change in the DNA if the DNA was the actual source of information or orchestration. And so, but what I'm finding as I look into this is that they don't know, they haven't been able to isolate all of those specific gene sequences. And so there's no way to actually prove or disprove that it's the DNA that's doing it. And now with CRISPR and stuff, you know, there's people out there saying, serious scientists that are saying that we'll be able to engineer any life form, humans included, to pretty exacting specifications. But even then, if you listen really closely to the talks, it's all a matter of probabilities. So we know that if we change this particular gene sequence, we have a higher probability of getting someone who's tall, or getting someone who's smart, or getting someone, you know, blue eyes is an easy one, but the more difficult ones are just going to be probabilities. And so, but then the idea of the morphic field comes in, and if that's the source of information and orchestration, then the DNA, what is it doing? It's actually being used by the morphic field to produce the proteins to make the body to do the functions. And then there's the whole instinct thing that, you know, classic materialistic science has absolutely no answer for. Anyway, I'll quit talking and let you talk.
Yeah, well, you know, it's I was just actually, I guess I've been thinking about this a lot because I'm developing a course on what is life, biogenesis, or the origin of life. And you know, a big part of that is how does biochemical reaction networks that are capturing energy from the environment and harnessing it to build macromolecules, organic molecules into large complex biological molecules. How does that go from being a abiotic process and transitioning into a biological one in which those chemical reaction networks are translated and encoded in digital chemical polymers, so that basically these chemical reaction networks form a hard drive, a memory, from which they can reconstitute the metabolic pathways for billions and billions and billions of years to come. But that transition is extremely perplexing, and it's actually one of the biggest questions in science. And then equally, the changes in that code are what are going to introduce novelty into the system, and hence evolvability and adaptability. And so we have the idea that comes from Darwin that these changes in that code are incremental and random. And basically, the organism is capitalizing on fortuitous changes, random mutations, to increase survivability, adapt, and evolve. Well, you know, even in current modern evolutionary biology, most biologists understand that that is not a 100% correct picture. That can't be how it's working. There has to be some degree of information exchange in the system that is directing this evolvability to a certain extent. And depending on where you fall on that spectrum, like myself, I think it's being directed by information exchanges to a large extent, where the rate of so-called like serendipitous mutations is very low. Because you use the example of a caterpillar. Imagine plants 130 million years ago, they didn't make flowers. Now insects were crawling on them. And then at one point, now the plants, they can't move around, but they want to essentially recombine their genomes to make new plants that are slightly different and can adapt and evolve and are a better fit to the environment. But they can't move around to mix their genes. But they can use the insects to do that. But so, if we're following this random mutation idea, the idea is that at some point, there was a random mutation that caused the plant to start secreting nectar at its seed production centers. And that attracted the insects so that it could more readily exchange the pollen and undergo reproduction via recombination. But I mean, the idea that it's going to randomly and accidentally start producing nectar at the site where it needs insects to come and interact with the pollen, it's like, okay, given millions and millions of years, maybe probabilistically that might happen. But I think actually a simpler explanation is that there's some amount of information exchange and intelligence involved, and that there was directed mutagenesis, right. And that flowers were evolved in a somewhat intentional manner. And it wasn't a mutant. A freakish mutant that led to that. Right. Now, the second part of your question, though, is can we show this? Now, other scientists are a little bit ahead of us on this because they have shown it, directed mutagenesis. And so actually, you know, the kind of core concept that most people are left with right now is the conventional Darwinian model, which I already explained, you know, that it's random mutations that sometimes are good, sometimes are bad, and sometimes the organism can capitalize on good ones, and that's evolution. And it takes millions of years and it's incremental. But the thing is that's not how you see it in operation. In fact, evolution can be punctuated by rapid onset of radiative adaptation. And you know, it's not really incremental or random at all. Now, so there's what's called the new synthesis, which is kind of the updated version of Darwinian evolution, which brings this intelligence into the system, which says it's not blind and random, that the biological system, the living system, at its heart, one of its defining characteristics is intelligence, intelligent behavior. And that goes all the way down to what it's doing with its genes and DNA. Because if an organism, you know, if we were to go back even billions of years ago, if an organism could figure out a way to increase the rate of changes in its DNA in a controlled fashion, that's evolvability, it would have a much higher success rate of survival. And, you know, going back to Darwinian evolution, it's all about who can survive and reproduce and pass on the genes. So, you know, even within a conventional Darwinian framework, we would expect that organisms would have evolved mechanisms to direct changes in the DNA and direct mutagenesis to have beneficial mutations. That's what I would expect from Darwinian evolution in the conventional theory. Now, the new synthesis has shown that this is absolutely what occurs. And there are already experiments that show this. So one of the most famous is with the LAC-Z knockout mutant experiment. So you've got bacteria, Escherichia coli. It likes to eat sugars. It likes lactose. If there's lactose in its environment, it'll secrete enzymes that break it down, and they can use it for energy and to make new Escherichia coli. Well, with things like CRISPR and other gene editing technologies, you can go in and you can knock out the gene that makes the enzyme that breaks down the lactose, the lactase gene, the LAC-Z gene. Now, Escherichia coli can't metabolize lactose. And so what some experimenters did was they knocked out the LAC-Z gene in a population of Escherichia coli cells, and they put them on a minimalist media, a media that had a little bit of glucose, like just enough that they could survive and slowly multiply, but it was very arduous for them. You know, it wasn't a good supply of food. So they were basically starving these poor little bacteria, but it had an abundance of lactose. They couldn't metabolize it because the experimenters had knocked out the LAC-Z gene. Now, what they found is that under repeated passing of these bacteria, they would much more rapidly than would be expected from pure random probabilities, and very quickly turn back on the LAC-Z gene so that they could get the lactose. And so in the extended synthesis and evolutionary biology textbooks, you'll find this experiment and you'll find the term directed mutagenesis. And so, you know, it's speculation. It's not really understood by most how this could happen, but obviously they are sensing their environment, saying, here is a food source that we want, we can't get right now, but if we change the nucleotide sequence of this gene, we can turn back on this enzyme and get that food source. And that's exactly what happens. Now, go ahead, Inés.
And maybe they remember, because in the morphogenetic field, they know that they can process lactose. So that would maybe probably, you know, be a source of information.
That's so great, because so we have an experiment to test exactly that. So one of the ways that they're able to do this seemingly very, very intelligent process of identifying the nucleotide sequence isn't right, changing it to the right nucleotide sequence to make a functional protein to get the LAC-Z. We have an experimental design that would test exactly that we can couple these LAC-Z mutants, knockout mutants. We can couple like a population of them more strongly to the morphogenic field, the unified space memory network. And if we were to run this experiment, I believe we would find that those groups that are coupled more strongly to the unified space memory network, they're getting information more readily from the morphogenic field, the morphic field. They're able to turn back on the gene that's needed much more quickly, much more rapidly. I could actually...
Because they're able to leverage the feedback feed-forward mechanism
And
by being in contact with the morphogenic field.
Not only that, but that's actually our main hypothesis in our paper, The Unified Space Memory Network, is that we are explaining evolution as a directed process that is occurring via information feedback feed forward with the entanglement nexus of the Unified Space Memory Network. And so the idea is that pretty much all adaptive evolutionary change, the information for that is being accessed via the unified field. If an organism is coupled more strongly to the field, it can undergo this kind of evolution much more quickly. But you can extend it to kind of explain the novel and serendipitous changes, seemingly serendipitous changes across evolutionary change history. So like even the evolution of bipedalism of hands, at some point aquatic organisms crawled up out of the oceans.
Mm-hmm.
This idea would say that, well, the changes for that, the information is present in the morphic field.
Yeah.
There's morphic resonance. And so they're accessing the actual gene blueprints and the morphogenic blueprint basically across time where it's already existing and it can function as a template. And so there's not really much randomness about it. You do still have a degree of randomness that's occurring, but it's a much more intelligent and directed process. We explain it as like a natural mechanism that these kind of changes are increasing the ordering of the system, of the living system, increasing the amount of entanglement across the system. And entanglement is non-local. So that means entanglement not only in space, but also in time. So lineages that have strong entanglement across time are going to survive and evolve. But basically, this hypothesis is a falsifiable one. We have an experiment to test it. We could prove it wrong, potentially.
We have to be open to that, right?
Yeah.
Science.
If we ran the experiment,
Yeah.
So, yeah.
Well, this is incredibly exciting that people such as yourself and other scientists are focusing on ways to prove that there's intention in the equation, right? And because, if you go back to that Rubik's Cube thing, where Nassim always used to talk about, where if you give someone a Rubik's Cube and ask them to solve it, then it would take them, 18 billion years if you didn't give them any feedback, probabilistically, of course. But if you gave them a yes or a no, it would take them 2 or 3 minutes. But the part of that little story that's interesting is the fact that they had a goal. There was a question put to them. There was a prize. There was something they were going toward, right? Solve the cube. And so if you just throw a bunch of organic material into a soup and it doesn't have an intention, it doesn't have a purpose, a place that it's headed for, then it's not going to do anything. It's going to bounce around randomly. And I think you even published a paper on this about early RNA molecule assembly and how there was the ways to do it wrong had like 8,900 zeros behind it or something like that. And the way to do it versus the way to do it right, which to me proves beyond a shadow of doubt with numbers that the advent of life on Earth was not a mistake. It couldn't have been. It's just flat out impossible.
Yeah.
And then you take that to the cosmological level and how are conditions just right in physics that we're here, right? All of the variables that if they were off just a little bit. And so, you know, people, physicists that are proponents of whatever theory invent 10 to the 500 other universes so that this is the only one. So that it can keep that random, unintentional thing to keep God out of the equation. But I love the convergence where this is a really important time where being a spiritual person, a non-denominational spiritual person, has really it's gaining so much more traction because of the work of science to actually get in and explain that yes, there is intention, yes, there's information absolutely everywhere tying us all together. And so what you were saying about if you could increase the connection between an organism and the unified space memory network, then it would be able to adapt better. Well, I think that on Earth, we as humans have this ability to evolve ourselves within the span of our own lifetime. And so if we can set our own intention and we can become quiet and become connected to that space-time memory network, then we can actually evolve much more quickly ourselves during our lifetime, right?
Absolutely.
And that evolution is the only thing that's going to save humanity.
Yeah.
Because it's kind of a shit show out there.
I actually have some work explaining how within a single lifetime you can evolve. I mean like changes to your genome, and not just like epigenetic changes. So obviously, you know, you can change the epigenome, which is just, you know, highly responsive in feedback to everything you're doing. But there's actually a mechanism in your body where it'll change the underlying nucleotide sequence. So I mean, you know, change your genome. So you know, it is possible to like have evolution within a single lifetime. And you know, I think the crucial aspect of that is, you know, how coherent are you? Like, are you coupling strongly to that unified field so that these changes that are occurring, where you're undergoing what are called somatic cell nuclear recombination, this is occurring in your brain in one of the places. There's other locations in the body where it's rearranging your genome. You know, if you are coupled very strongly to the unified field and maintaining strong, coherent connections, those changes are probably very beneficial. And, you know, those cells go out and propagate and populate your body. So, I mean, I just, you know, it's, you had mentioned that, you know, evolving in a single lifetime. And that's, I think, literally possible.
I would like to add, because it also would depend on what part of the field do you couple to. And that made me think, because of course, in the field, just like Nassim says, that you tune your heart to the frequency of the field, but depending on the frequency, you can tap into fields that are not so much, that bring different information. So the information depends on the frequency you tap into, basically, no? So for instance, I have thought a lot about aging, you know, because, you know, we don't want to age. Our life is like too short for everything we would like to live and all of that. But I realized that there's some kind of coupling to this memory field in which people age because that's what they've used to see. You see people aging all around, so you age as well. You follow the flow. You follow the morphogenetic field of the memory.
Mm-hmm.
And so you really have to be conscious to what part of the field you're coupling to. because I'm sure that aging, of course, there's this biological change as well in the telomeres, but that's a feedback-forward loop. So if you're conscious of that, you can maybe hijack that and so make it, you know, stay longer and not so much. But I know that because for me it's really when you stop seeing someone for a very long time and then you suddenly find them again and you find their resemblance with their parents, of how much they have aged and how much they look like their parents, that made me think immediately that we do that all like by inertia, like, okay, we have to age. Yeah, everyone's aging, let's do the same, you know. So there's
Well, so
something
now
to
you're
that.
well, so then now you're talking about worldview and belief systems,
Yeah,
right? And the
exactly.
impact that they have on the expression of our genome and our morphogenic field. And and William, what you were saying is that if you can actually change a nucleotide sequence in your DNA stream, now we're talking that it's accepted in science that would be heritable.
Oh, well, no, not necessarily,
I guess if
because
it changed the right one, if it got into the sperm and the ovum, right?
yeah, but you know, actually, to my knowledge, this mechanism doesn't go to the gametes, but that certainly doesn't mean that it doesn't. It just means that I haven't seen a study on that yet. And actually, now that I'm thinking about it, I wouldn't be so surprised if it did. But you know, one of the things about the reason why the mechanism is called somatic cell nuclear recombination is to distinguish it from gamete cell nuclear recombination. So the recombination occurs in gametes constitutively. You know, so if you were to have this kind of change, they did propagate to the gametes, it's going to be recombined again. So, you know, I don't know if the change would necessarily be passed on. You know, there's definitely, though, I think, certain levels of that information of what you've done to evolve to a higher state, let's say a more coherent way of being, that are passed on and heritable in that sense. But yeah, it gets a little bit tricky with the gametes because they undergo reprogramming. So they've got enzymes that come and remove all the epigenetic patterns from them and put on a blank slate, a new one. And then, you know, they undergo recombination where all the chromosomes are rearranged and shuffled around. So you know, they kind of get a good amount of, I don't want to say scrambling, but diversification, the diversity introduced in. But certainly, you know, I'm sure that there are mechanisms by which that kind of information is passed on and is somewhat heritable, because they've shown that, you know, even with epigenetic reprogramming, you know, offspring still can carry like an epigenome blueprint of the parents that the parents formed during their lifetime.
Like the grandmother effect. Yeah.
Mhm.
Yeah, well, so, and that I think brings up the idea that, I mean, we've been talking about, when we talk about the nucleotides and the gene sequences, we're talking from a perspective of DNA is the prime determinant factor in how you express as a human being, right? Of all of your physical characteristics, all of your capabilities, all of your instincts, all of the function, right? And my understanding is that we don't actually, that's not actually true. That the DNA is the blueprint for the building blocks that we then use to create a human being, for instance, right? And there's something that is responsible for the orchestration and the information of how we're going to use those building blocks to create a human being, and that's what we would call the morphic field. And so, but you know, isolating variables in the morphic field is not quite as easy as isolating the variables in a gene sequence. And so so when we talk about, is it heritable, I think it makes sense if it's the morphic field that even though the gene sequences were, brought back to original state and then resequenced or jumbled around, that the actual morphic field that's using them, if the change is there, then it's heritable anyway, and we see the grandmother effect.
Yeah, that's a good point. Yeah, and certainly the DNA more than anything, it's a responsive part of the cell. It responds to cues, information that's being sent to it. It's not like the commander that is sending out commands. It's more like it's responding to commands and replying. It's like it's much more of a responsive element than a controlling element, if anything.
Just out of curiosity, how far into your education did you learn that the central dogma is bullshit?
So actually in my when I was doing my master's degree, I was taking classes on complexity theory in biology, and you would see like the morphogenetic diagrams for the development of an organism, and when you look at these things, it's just this complex network where you can't hardly pick like a single line going from point A to point B, because there's all these intersecting interactions and bifurcations, and so it's a holistic system that to try to apply a reductionist method to it is just fruitless. I mean, and so it's like very much apparent that there's like a field behavior that's occurring here that is more of the orchestrating element is a field type dynamic, but it's definitely during my studies for my master's degree, because that's also when I started to investigate and research things like mobile genetic elements. And so if you look at those, and so like one of the questions that I was investigating, one of the thesis that I did my master's on was, okay, so humans have 99.8% genetic identity, gene similarity with chimpanzees, which are supposed to be our closest related cousin species. So how does that 0.02%, 0.2% difference give you a human versus a chimpanzee? So and then so like I went and did some research on this, and well, it's obviously it's not in the gene coding sequences at all. I mean, not least of which because the gene coding sequences, conventional gene coding sequences, only are a very, very small fraction of the entire genome. And if you look, about 50% of the genome are satellites or microsatellite DNA, which is extremely interesting. And it's extremely interesting because it doesn't have a digital code in the linear sequence of nucleotides, because another name for this satellite DNA, because it's satellite DNA because you'll find it on the periphery of genes, is tandem nucleotide repeating elements. And so it'll be like the string of up to like 800 repeats of like TTGGG, and that'll just repeat. Now, if it's just a repeating TTGGG, that doesn't code for proteins. So it's not a coding gene. And so what is it doing? And why is 50% of the genome these non-coding segments? Well, a few interesting things about those is that if you want to identify a species, you don't look at the genes because the genes won't resolve any species because they pretty much all share the same genes, and there's very little changes in them. I mean, especially like if you want to try to, if you had a DNA sample and you wanted to try to say, is it a chimpanzee or a human? You didn't know. You wouldn't look at the genes. You'd look at these repeating sequences. And actually, that's what produces the so-called DNA fingerprint. So, but that's kind of telling you something. So these are producing the DNA fingerprint that is the distinguishing characteristic, allowing you to resolve the species as distinct from another one, and actually allowing you to identify a person from another person. Maybe this has something to do with the individuality, with the species level distinction. And so one of the interesting things is, so it doesn't have a linear code. There's no information in linear nucleotide sequence, but if you look at the spatiotemporal patterning, it gets extremely interesting because there you do have potential information encoding in a spatiotemporal pattern. So like that TTGGG doesn't code for anything, but in a spatiotemporal sequence, it forms a G quadruplex of the DNA, which is a 4-stranded DNA structure. And this regulates the genes. And so this is why you find these regions on the periphery of genes, is because they're regulating the gene expression. They're changing how those genes are expressed.
So they would be modulating and or perhaps even radiating a certain pattern based on their spatiotemporal configuration?
Yeah. And so, you've got direct physiological modulation of the gene expression via these spatiotemporal patterns, but also the electron orbital configuration of the DNA, when it gets into these different configurations, you're going to have different resonances, different resonant frequencies. So, it's going to have different frequency of photon emission, electrical conduction, all of these kind of things. And that's where I think you're having some of the strongest coupling with the unified space memory network, the morphic field, is via these resonance structures, which when we look at it, we don't see a code because the code in this particular instance of the DNA is a spatiotemporal configuration pattern. This is, and so that's where you can start to get into where, well, so those are passed down to the progeny. So maybe that's where, like, some of the experiences that you've had in your own life, those are passed on through the gametes into your progeny. And, that's one of the reasons why, you can do a paternity test, is because that DNA fingerprint is going to be very similar. And that DNA fingerprint is those non-coding regions, so that is being passed on. So maybe, I think that's where you have some of the strongest morphic resonance, morphogenic coupling is in those non-coding sequences. And then, so that's 50% of the DNA. And then like 40% of the DNA is mobile genetic elements, endogenous retroviral elements. And so that gets into like a whole another thing. And when you take these two into consideration, I mean, it's just that the majority of all evolution is not random. I mean, these mobile genetic elements, horizontal gene transfer, RNA to DNA editing of these mobile genetic elements, all these hundreds of thousands of regulatory mechanisms, none of them, those are not random, so very quickly when you start delving into the actual molecular genetics, it's like, oh, okay, so this idea that things are chugging along and you get a little random DNA mutation, and over 1 million years, you get enough of those that you get a mutant that's beneficial. That becomes BS very quickly,
Yeah. Out the door. Well, and everything you're describing suggests that that there's changes that are happening to the DNA, and that is happening from a field, right? From some sort of field dynamics based on the information exchange. And if there's something happening to the DNA from the field, and that means that I can change my DNA if I can really get in touch, if I really find that Bindu point, then I can and come up with an intention that resonates with it, then I could probably make that change in my DNA. And also, someone else was talking earlier about if I do that, then that paves the way for the rest of the species to do something similar.
Mm-hmm.
Right. And I don't know what the science is behind that, but that seems to be the way it goes. That talking to you makes me smarter.
Oh,
And talking to you makes me smarter.
you know what about that? I would like to add something. Part of the experiment in the how do you say that? Okay, so there's well, you know, there's been many experiments that were hidden, not like by CIA and all things like that, you know, remote viewing and all of that. But at that time, and like so many years ago, people who have been working in these secret programs, they have been also looking at telepathy, but also one of the things that the training that we were that does the bond tradition and all of that is brain to brain transmission of information. So apparently you can send packages of a huge amount of information that it's sent by certain initiatic methods. Okay, it's not like so easy, but once you have trained the other one to receive the information, then he can get it, and this information unfolds in 3 years. So let's say you're a master and you take and you have all this knowledge coded somehow. I don't know how they do it, and then they just choose one. At the time, at many years ago, that was like the case. You choose one. Look, they had the mentor and the master, and he would transfer the package, and that package would unfold in 4 or 5 years. I don't know. It would take some time to unfold, and so that's exactly what you're saying. It's like that is already we know that, and that's things that we want to also experiment, but that's something that we can explore, and it's already known by many cultures already. It's just
exciting.
Yeah,
Very exciting time to be alive.
Yeah, very exciting. Yeah,
excellent. Well, thank you for that, and thank you, William, for the answers and the conversation. It's always so rich and enlightening.
Yeah. Okay, so Gabe, maybe we have time for a little last question because it's almost 2 hours, or not maybe. So there's here, this is a question about vaccines. Okay, Maxine Hamilton Stubber. So she's asking, oh, where's William? We lost William. Okay. Okay, so let's wait to see if he's coming back soon, because that would be a question for him to respond. Oh, William, hi.
I am back. Sorry about that.
No worries. Okay, so here's a question about if you're aware of any direct effect of the vaccination, of the COVID vaccination. Yeah, about that, if you want to answer.
So do I know about any effects, like negative effects from the COVID?
Yeah, vaccine? Like what is
well, so yeah, you know, I'm not exactly sure what you're allowed to say on this kind of stuff, because you know, if we're live on Facebook, this will get taken down if I say the wrong thing. You know, so
so maybe just don't say anything.
I would hate to have the video censored, because you know, I mean, I can't say kind of more generalistically that you know, I have, I guess, what you could call a much more nuanced approach to the vaccines. You know, so as a molecular biologist, you know, I'm very much familiar and studied in the field of virology and, vaccine technology and all of that. But one of the things is that
the question is effect of vaccination on centrosomes.
Oh,
Centrosomes.
oh,
Yeah,
okay,
That's the
all right.
particular question, yeah.
Well, on centrosomes, there's not going to be a direct effect on the centrosome from if we're talking about the mRNA vaccine or even the spike protein vaccine in the current COVID vaccines. Yeah, there's not going to be a direct effect on the centrosome that I can think of anyways. Yeah. Yeah, I really don't know what you're allowed to say about that kind of stuff without getting kicked off of YouTube and Facebook. And all that. I mean, because I've seen folks who I think I've seen make just totally relevant, unbiased, level-headed observations, points about vaccine technology, and they get huge backlash and censorship, and so it's just the fact that it's more of a political discussion than a scientific one kind of brings me some hesitancy on how freely to speak about it.
That kind of answers the question.
Oh yeah. Oh, okay. Yeah.
Yeah. Okay, so maybe we can wrap the call here. Yes, it's been 2 hours, so yeah. So last words. Goodbye.
Thank you everybody for joining us. It's been a great discussion. I just always thoroughly enjoy the insights and inquiries that are brought to the discussion from our community. It's just, I love these areas of exploration and thought that we get into. It's fun to explore. I hope the same for you. And we'll definitely hope to see you on some of our next faculty Q&As and calls, and we can get into some more unified physics and unified science discussions.
Yeah. Okay, so on my behalf, thanks for being here, always supporting us, following the course and disseminating this information. As William, I'm very happy to be here in exchange these moments with you. It's really, really something that I love to do. And so we hope to see you soon in the Q&A, maybe probably the next Q&A. So goodbye.
Thank you all.
Thank you all.
Bye.